Okay, so it is 11 o'clock, and I thought what we would do today is to start promptly at 11, picking up questions that are left over from yesterday, and then after about 10 minutes or so, we'll shift to what was planned for today's lecture. So there were two things in the chat. One was that somebody was curious about having a video of the RNA polymerase walking along DNA. If you poke around the internet, I think you can find things of that flavor, which are cartoons. One, of course, can't directly visualize this, but you can also look at the experiments where you're manipulating single molecules, and you can see the traces of the position of the RNA polymerase along the DNA as a function of time, and you can see discrete steps and so on. And so these are pretty classic experiments that you can Google should be sufficient. The other question was about the origin of experiments where you can finally measure concentrations of proteins accurately inside cells. So what we're doing is every single one of the references that you see in the slides, they will be in a file where you can find them if you follow the links on the page for the course. If you go back, the slides, of course, themselves are also posted. So if you go back, you'll see that there are references to papers by Gregor et al. For the measurements in the early fly embryo, and you'll be able to find those papers by following the links. So I hope that will be sufficient. Maybe there are other questions left over from yesterday. You can either type something in the chat or raise your hand. Yesterday, it seemed like people had more questions, but maybe on reflection, they've settled or something. Yeah.
I did have one question.
Yes. Hey, Tom, how are you?
I'm well, thank you. Great to be here. Which is on the precision protein measurement. Is there? You said this was you could worry about lots of things. I have been worrying about one thing, and you can tell me why I shouldn't worry about it. Which is two ways to think about it. I've measured a molecule by diffusion. It wanders away, it comes back again. It's out of my cell, it comes back. And diffusion being what it is, has high statistics for return probability.
Mm-hmm.
I think that's the same as saying I can sustain local fluctuations in numbers that scale over a number of different length scales, like long time tails, and I might get wrong by suffering some of those fluctuations in my neighborhood.
So this is a great question. The version that so there are many versions, there are many ways in which that question becomes relevant. One is that if you want to measure a diffusion constant, you have to be very careful. Another is that the statistics of diffusion are very different in different dimensionalities, of course. And so, for instance, the Berg-Purcell argument that I gave yesterday is explicitly an argument about 3 dimensions. You will discover that if you try to make that argument, well, 2, of course, is marginal. If you try to make that argument in 1 dimension, things get very weird. And in fact, somewhere in the argument, you need to say something about the size of your detector. And so it's natural to think that if the way in which the cell is sensing concentration is by having the molecules bind to a binding site, then the size of the detector is the size of the binding site. At least that seems natural. Could be wrong, but that seems like a good place to start. But we know that proteins that bind to DNA can find their way to their binding site by binding. Let me see if I can do this. It's hard enough to do this if we were actually together. It's even harder to do it in the camera. So if this is the binding site they're supposed to find, my arm is the long piece of DNA, then the protein can bind to the DNA and diffuse in one dimension along the DNA until it finds the binding site. And there are examples in which we know for sure that this is important. People immediately made the argument that as a result of that, the detector is actually much larger because you can bind anywhere and slide along, and then effectively you're that much larger. The problem with that argument is that the statistics of motion in the one dimension is very different than the statistics of motion in the 3 dimensions. And so you have to do a much more careful calculation. And when the dust settles, it turns out that the effector detector really isn't that much bigger. But that actually depends somewhat on the parameters of the system. So I think, I doubt that the examples that I just gave you exhaust the places where one should be more careful in thinking about diffusion. And for example, we know that diffusion of, so if you think that an important step in the process is that the place where the transcription factor binds and the place where the RNA polymerase starts reading have to come together in space by the DNA itself bending, the chromosome bending, then we know that the motions of the chromosome are subdiffusive, not diffusive. And that could simply be for good old-fashioned polymer physics reasons, or it could be more interesting, or not. I mean, we don't. So, yeah. As many of people in the audience will know, there is a simple polymer physics. If you do the problem of an elastic string, and you ask how does one point move, the answer is that it doesn't diffuse; it subdiffuses. Right? So the mean square displacement grows not linearly in time, but as a smaller power.
Yeah. And even in a non-Markovian way. And it's really
right,
subtle.
Right, the only way that's possible is if there are long-time correlations between the motions. Yeah. And so just the fact that the object is extended means that diffusion is more complicated. Now, you're sort of saved by the fact that in the Berg-Purcell argument, it's not the diffusion of the DNA that matters, it's the diffusion of the transcription factor, but there are plenty of reasons to worry. And so the whole issue of, how do I redo that calculation in a complex environment, right? Where, for example, so for instance, when you observe a diffusion constant, the diffusion constant you observe is typically measured on a length scale of a micron or longer, because you're looking under a microscope. But the diffusion constant, which matters in some sense, if you take the picture seriously, is the diffusion constant on the scale of the detector, which is at least an order of magnitude smaller, probably 2 orders of magnitude smaller. So there are things that can happen between those scales. And the question is, how, can I plug in to my estimate of the noise level the diffusion constant that I measure? Or is it some bare quantity that matters, which is hidden from me? Of course, concentration, the concentration which enters should be the free concentration of the stuff that's diffusing around. And many of the mechanisms that slow down diffusion slow it down simply by having the thing that's diffusing stick somewhere, in which case it's not free. But then when you measure on a micron scale, you can't distinguish the free from the bound. So there's a set of subtleties here, which if you want, I think are worthwhile. I don't think I said anything that will be severely disrupted by thinking about them.
I don't.
But I
think so.
also don't think there's a single reference that lays it all out clearly.
Oh, that's interesting. No, I just think there are some really interesting subtleties to the first passage problem when you're looking at, say, the end of a chain, because of that non-Markovian stuff that
So there's a
has
very
bad
nice
view.
review article by Olga Dudko and Yaojun Zhang called First Passage Times in the Genome or something like that.
Okay, all right. Didn't know about that. Good.
So let me actually see if I can remember how to make a note here. There's something about first passage times.
Mm-hmm.
In the genome, I'm not sure if that's exactly the right title, and that is Dudko and Zhang. And it's relatively recent. Good, there are some questions in the chat. One question is about paths into the subject. What other disciplines in physics give you the greatest capacity to understand physics life? What I would like to do is to save some time at the end of today's lecture for a kind of general discussion. So maybe we can get back to this question then. And so I will ask Mr. or Ms. iPhone to remember their question. And then John Bastola asks about whether the examples, so let me generalize the question a little bit, which is that in the interest of trying to illustrate an approach, I choose examples. And these examples are chosen both, they're chosen in the sense that they're the ones I choose to lecture about, but as you realize, these are also problems that my colleagues and I have worked on. So the question is, how general are the things that we find? Let's say that maybe that's the best, the most encompassing version of the question. In physics, we're used to the idea that you sometimes go in a corner where things simplify. And in that corner, you find things which you are confident will be general, despite the fact that you looked in a corner. In biological systems, we don't yet know how general things are. It's always true that things are understood by looking at places where the question is simplest and clearest. So it's particularly true in biological systems that it's very easy to find yourself in a place where if you want everything's happening at once. And so it's hard to get a grip on any question in particular. So I think the truth is that we don't know. And the challenge is we're still in a phase where we're collecting examples in the hopes of bolstering a view, right? And then you have to decide at what point do you have enough confidence in the view that you say that you've learned something. Let me point out that for example, the fruit fly embryo, I mean, it wasn't our idea that the fruit fly embryo is a good place to look to study the very first steps of development. That's a very old idea that goes back to very classical biology over a century. And there are many ways in which development in a fly is completely different than development in other organisms. And yet, much of what we've learned, I mean, parts of what we've learned have proved to be very general. And so even within biology, sort of within the, you know, let's say biology as practiced by biologists, right? There's biology, the literal meaning is the study of life. And then there's biology, the academic discipline, which has a particular approach to problems. So biology as practiced by biologists, within the academic discipline, there are already questions about generality. And you know, obviously, at some level, everything's different. And in other useful ways, things are very much the same. So it's easy to set up the dichotomy that, you know, biologists want everything to be different and physicists want everything to be universal. And I don't think that's fair to either side of the discussion. Physicists are perfectly capable of appreciating variation, and you could argue that, for instance, molecular biology as a subject would not exist if it were not the case that there was a tremendous amount of universality at the molecular level. So most of you by now, if for no other reason than listening to the news about the pandemic, know that there's something called the polymerase chain reaction, PCR. So if you want to detect the DNA, if you want to detect a small piece of DNA, what you do is you make many, many copies of it until you have lots and can do chemistry. And the way you do that is called the polymerase chain reaction. The reason this all works is because, so in order to do the polymerase chain reaction, what you need to do is to, right, when you copy the DNA, you pull the two strands apart, and then you copy each strand. Now, normally this is a very complicated process in which you have to unwind things, and you have to sort of unwind, open up, and copy all at the same time. And but there's an easier way to do it, which is imagine that you could just warm everything up, and then the two strands fall apart because the bond isn't strong enough to work at high temperatures. And then you let a polymerase run over the sort of opened up strands, and then you lower the temperature back down, and now you have copies. But in order for that to work, you need an enzyme that works at very high temperatures where the strands are already separated. So you don't need all the other machinery to do the separation. So the way you do that is you go down to the vents at the bottom of the ocean where there are very high temperatures, and there are bacteria that live there, and they, of course, have an enzyme for copying their DNA, but it works at high temperatures. Also high pressure, which is why everything stays together. But then you can take that molecule and use it at high temperature to copy human DNA or viral DNA. These are things that that molecule has never seen in its evolutionary history, and yet it works. So there is this interchangeability of components that makes molecular biology work as a subject, which is a kind of universality. There is, okay, so the last question, I think questions are getting a little more philosophical, and so maybe what I will do is start today's lecture, and we'll return to these more philosophical questions at the end. So today, and we have once again the problem that advancing the slides is somehow problematic. So today the plan is that we're going to talk about emergence, and the example that we're going to use is from networks of neurons. And what I'm going to do at the beginning is so the first half of the day will be sort of up to the break around noon. We're going to talk about simple models for neural networks, which has a fantastic history. And then we're going to talk about, and in particular, the study of simple models for neural networks goes back long before we could collect the kind of data that could really test many of these theoretical ideas. And so I want to give you the theoretical foundation, then I want to tell you about progress in being able to do experiments, and then we'll get back to seeing what we can accomplish as physicists to talk about emergence in these contexts. Okay, so let's see how this works. So the history of simple models for neural networks goes back at least to this paper in 1943 by McCulloch and Pitts, this sort of magnificent title, A Logical Calculus and so on. And what they showed was that if you imagined that what neurons did was to add up their inputs, so there's one really important fact you need to know about neurons, and that goes back to the early 1900s. And that is that when you measure the voltage between the inside of a nerve cell and the outside of a nerve cell, that voltage is not zero. The cell's working very hard to maintain a voltage difference between the inside and the outside. And when the neuron is active, that voltage changes. And by tickling the cell very gently, you can cause the voltage to change by small amounts. But once you push the system enough, the voltage gets triggered, and what comes out is an action potential or a spike. So there's a voltage change that lasts for a period of order of a few thousandths of a second, a few milliseconds. That voltage change is very large. It's of order 100 millivolts across the membrane. And when neurons communicate with each other, they can communicate over long distances because the cell has a long process called an axon, and that pulse propagates along the axon. And we understand molecularly how that works. It's a beautiful chapter in the physics of biological systems to understand all that. We're not going to talk about it. The only thing you need to know is that there is this all or none behavior of individual neurons. They don't generate half action potentials. So what's happening when the action potential is generated is that the conductance or resistance of the membrane is actively changing. And since there's differences in the concentration of ions between so the reason cells are electrically active is because they have pumps that pump ions in and out. And so if you measure the concentration of ions, it's different on the inside and on the outside. So what this means is that if you just open a hole in the membrane, some ions will move one way and some ions will move the other way. So that's how you get currents flowing. The membrane itself is largely a resistor. It has a high resistance and is largely a capacitor, actually. So the point is that this business of opening holes doesn't just happen at random. There are specific molecules that live in the cell membrane. Those are also proteins. And they open and close in response to various signals, including the voltage itself or the electric field across the membrane. And the dynamics of that process where you inject current, that changes the voltage, that causes some channels to open and some channels to close, which changes the amount of current that's flowing and so on, can produce the sort of nonlinear behavior that you need in order to generate these stereotype pulses. Beautiful subject, not for today. What McCulloch and Pitts said was, let's imagine that what neurons do is add up their inputs and take a threshold. And so that thresholding is like the generation of one of these action potentials or spikes, okay? And what they then argued was that if you connected these things to each other, you could perform various logical operations. So taking the threshold, you start thinking about things being binary, right? Or remember, this is 1943. So if you think about the history of computing, there were no devices around that worked in binary. So what they said is that what a single neuron does is a logical operation. It takes inputs and says yes or no. And then you imagine that you connect these things to each other and you can set them up so that they produce more and more complicated logical functions of the inputs to the system. And eventually, you can argue that they can compute any logical function. And so that means that by just making a cartoon of what neurons are capable of doing, you can imagine computing almost anything. So that was a very beautiful idea, and actually had a big impact on the early days of computing. So there's a path from McCulloch and Pitts to the early work of von Neumann and so on, and trying to build actual computers. And of course, this is sandwiched in between Turing's work on universal computation, which came before this, and the work on actually, building programmable machines, which comes later. Precisely because things are so universal, it's hard to draw conclusions. And so a guy named Frank Rosenblatt had this idea to make a much simpler model in which instead of the neurons being connected to each other in all sorts of complicated ways, they were connected to the basic idea was to think about what one neuron could do with its many inputs. and then to connect neurons together in a way where information just flowed from one layer to the next without lots of feedback pathways. And those devices, that model in the beginning was called the Perceptron. And you might be interested as a historical matter. Rosenblatt's original description is contained in technical reports, which weren't published. There's eventually a book. But the first references in which you can really the first journal articles in which you can get an account of this actually appear in reviews of modern physics. So the business of neural networks has its roots in the physics literature very deeply. If you look at the structure of these models, so there's again an interesting history. What happened? I mean, these dates are 1962, right? So why is it that we think of neural networks as being a modern subject? There are lots of interesting meanderings that happen along the way, which I won't review, but let me emphasize that these models, these perceptron-like models, are scaled down versions of the deep networks that are revolutionizing AI today. So there's no question in anybody's mind that what you see today, for example, the kinds of things that David talked about in his lectures, or that Viviana made use of in her lectures, they have their roots in this work from the early 1960s with the perceptron. In the early 1980s, there's, again, I'm skipping things, but there's this remarkable paper by John Hopfield, who went back in some sense to the model of McCulloch and Pitts. And what you could say is that everybody always is studying the model of McCulloch and Pitts. And then the question is, how do you structure the connections between the neurons? And I'll explain this in more detail. And one way to simplify the connections is to make sure that there are no loops. Everything flows in one direction, and that's the perceptron. And the other way to do it, to make things simple, and of course, that's not an accurate model of the brain, but it's a simplification, so maybe you can hope to understand something. And then the other way to simplify things is to make sure that there are loops everywhere, and they're symmetric. So this says that if neuron number 1 gives information to neuron number 2, neuron number 2 does not give information to neuron number 1. In this paper, there's also a simplification, and it's exactly the opposite one. It's that if neuron number 1 feeds neuron number 2, then neuron number 2 feeds neuron number 1 with exactly the same strength. And I'll explain this in just a moment. And all of these ideas sort of flowed outward in interesting ways. So let me now switch and try and do this, try and explain a little bit what's happening in these models. Okay? So again, you should think of a single neuron it has one side, which are called dendrites, and information comes in. Well, dendrites, and the other side, which is called the axon, where information flows out. And if you were to monitor the voltage here, the voltage difference between the inside and the outside, you would see that it's quiescent, and then you get a little spike, and it's quiescent, and a little spike, and maybe you get another one quickly, and so on. And as I said, these are on the order of 100 millivolts and a millisecond long. What's important is that, and the scale here is that the body of a cell might be, so in the center, deep in your brain, the body of a cell might be 10 microns across. So this thing here would be 10 microns across. Now, what's important is that if I have another neuron, let's say one sitting over here, and it has its dendrites going up, it can have its axon come, and it might branch and go in different places, but in particular, it can make a connection here. And this connection is called the synapse. And to give you a feeling for things, so what this means is that, sorry, just to get this clear, right? If this neuron is active, let's say this neuron is I, and this neuron is J. So here too, you have these action potentials streaming along. And so what's going to happen is that when the action potential arrives at the synapse, some interesting chemistry happens. Again, all of these are fascinating subjects with an interesting interplay between physics and biology. So what's interesting is that the way in which information is propagated over the long distance is by this electrical pulse. But then when it gets to the synapse, you get this funny conversion from electrical signal to chemical signal back to electrical signal. So what happens is that the arrival of the action potential triggers the release of balls of a small molecule, which is called the transmitter. So examples of transmitters are acetylcholine. That's the one which is most common between a nerve and a muscle, but also glutamate, the amino acid, and there are many others. And that small molecule diffuses across the little space between the two neurons and binds to molecules on neuron, right? It's released by neuron I and it binds to special molecules in the membrane on neuron J. And when that binding happens, those molecules open up and allow current flow into the membrane. So you go from the electrical signal of the action potential to the chemical signal of the transmitter back to an electrical signal of current being injected into the cell. And so there are other kinds of synapses, but that's the simple one. And so now the question is, how do you describe the dynamics of this system? Well, we know actually an enormous amount about the molecules that are responsible for generating action potentials. We know an enormous amount about the molecules that are responsible for opening and closing when the transmitter binds to them. We know a fair amount about the molecular details of how the transmitter is released. And so you could imagine writing an extremely detailed model of how this all works that's faithful to all the molecular details. And there are many cases in which that has been done, and it's really interesting subject. But if you want to understand, but it's complicated, right? And it's complicated in ways like we talked about before. There are many different kinds of channels in the membrane that are opening and closing. So a typical neuron in your head might, so your genome has the genes for more than 100 different kinds of channels. And a single neuron might make 7 or 9 of them in some complicated combination. And furthermore, they might distribute, the cell will distribute them differently, that the channels that are in the neighborhood of the cell body are different than the channels which are distributed along the dendrites, which are certainly different than the channels which are distributed along the axon. So if you want to give a realistic molecular description, it's going to get very complicated. So what many people did, starting with McCulloch and Pitts, was they said, let's simplify. The key thing is that at a single moment, the neuron is on or it's off. And on might mean that it's generating an action potential, and then off would mean that it's doing nothing. And because you've made things discrete, you've implicitly discretized time as well. So you're assuming that everything operates on some discrete timescale delta t. Okay. Now, clearly, this is too simple. You could think about other things, but as we'll see, the models of this flavor are enough to aha, I'm already falling behind on questions. So let me see if I can catch up a little bit. You don't tend to see very much, so there's a question about information propagating the other way. You don't tend to see signals propagating backward along the axon, just because of the dynamics, right? The signals start, right? The current is being injected into the cell at this end. So it's not too difficult to imagine that the signal is going to propagate along the axon this way. What is true, however, is that you can get information propagating backward along the dendrites, because that's not, because the dynamics are different. And that's actually important because that means that this synapse can find out whether neuron J decided to generate an action potential or not, right? So if you think that the action potential doesn't start until signals get down to here somewhere, then the synapse doesn't know whether an action potential got generated or not. It knows that an action potential arrived from neuron I, but it doesn't know whether whether neuron J actually generated an action potential. Because information can propagate backward along the dendrites, you can get access to that information. And that's important because the question of whether neuron I contributed to generating an action potential in neuron J is a question that's important for learning, right? So if this pair of neurons is embedded in some big network supposed to learn how to do something, then you somehow need access to information about what's causing what to happen, or at least what's correlated with what, so you have a chance of saying something about causality. And for that, this back propagation of information is important, but let me leave that aside for the moment. There are many different kinds of transmitter. Yes, Wikipedia will do that for you. Is there a refractory period? That is part of the reason why it is related to the fact that you won't see information propagating backwards along the action potential, that when an action potential propagates, it leaves behind it the membrane for a certain time can't generate another action potential. And again, we understand that in terms of the underlying molecular dynamics. Why does discretizing signal force discretizing time? Very good. Sorry, somebody also points out that we have a problem with the screen not updating. So let's try that. Does that work? It is, if you're thinking that the neuron is either on or off, you could think about there's some probability per unit time of making the transition between being off and being on, and then time can run continuously. But if you want to think about a deterministic model, then in some sense, right, since the transitions are discrete, you have to, it's easier to think about it as running in discrete time steps. I actually don't think this is that big a deal, although we could have a longer discussion about it. And yes, there's no magic about signals propagating one direction along the axon. If you inject current at one end of the axon, you can make the signals go the other way. But that's not what happens in the cell. So if you think that what cells are doing is basically turning on and off, and at the sort of at the finest level, you might think that that means generate spikes or don't generate spikes. It might be that you're having a slightly more coarse-grained view, and you're saying, well, the neuron is active or it's not active, and maybe that reflects more than one action potential. But let's sort of abstract away a little bit and think about it as being active or not active. Then that means that I can define a variable sigma I at t, which is 1 if the cell is on and 0 if it's off. And so then I need to write down a dynamics. So what's the simplest dynamics I could think about? I think about the time, let me choose units in which the time step is 1. So I say that the neuron at time t+1, well, what does it do? It's going to decide whether its inputs are above or below a threshold. So what it's going to do is take all its inputs, and we're going to say that it sums up the inputs with some weights, right? So at time t, each neuron sigma j is either on or off. Its neuron j could be connected to neuron I, and it has some connection strength, which we'll call W, and that connection strength lives inside the synapse. And then if this is above some threshold, I can take the step function, right? H of x versus x is like this, right? I can take the step function and say, well, that's the state of the neuron at the next time. And roughly speaking, this is what McCulloch and Pitts did. In 1943, and people have stuck with this. Obviously, this is too simple, but already McCulloch and Pitts showed that if you take neurons like this, you can compute arbitrarily complicated logical functions. If this picture is true, all of right, the only really interesting structure, I mean, of course, there's the structure of the thresholds, but if you think about it, if you could say that every neuron has a threshold equal to 1 and absorb the threshold into the scale of the weights W. So really, all of the interesting behavior of the system is contained in this matrix Wij, which are called the synaptic weights. And so in principle, these are arbitrary things, but then it gets complicated. Another possibility is that they are feed forward. So what that means is that if Wij is not equal to 0, then Wij is equal to 0. Right? So if I have a neuron sitting here and information goes that way, it does not go this way. And actually, more strongly, if you really want it to be feed forward, you need more conditions because you should avoid things like this. So if you want to build a feed forward model, you should make sure that all of the neurons send information through layers. And then they can go up to another layer. And so on, but the information never flows back. And so these models are the perceptron. And eventually, so the perceptron started with just one of these layers. And then people started to talk about multi-layer perceptrons. And then eventually, you get to deep networks. And so this is the state of the art of modern artificial intelligence. And it is still, in some way, the model that McCulloch and Pitts started with. There are some details which are different, which are important, but not hugely so. I mean, certainly not at the outline level. So you can trace what's happening today all the way back to these early ideas. And one way to think about it is that the arbitrary case is just too hard to say anything about. So in order to make any progress in understanding what these networks can do, you have to simplify somehow. So then the question is, what's the other possibility? The other possibility is the symmetric case, which was what Hopfield did. And it turns out, let me for the simple case, sorry, I've already slipped. Some of you will know why I did that. Tempting to do it again. Let me, for simplicity, put the thresholds at 0. This is not important. We can go back and do the more general case. What Hopfield realized was that if you have this model, and again, this is the McCulloch-Pitts model, just with, as somebody called it, the giant step backward of assuming that that the matrix W is symmetric. There is then a quantity E that you can form, which is the sum of sigma I, sigma j, weighted by the weight wij. And under these dynamics, E decreases. So it is something which is called the Lyapunov function. Right? So in general, if you have a dynamical system, if a Lyapunov function exists, it means that the dynamics always cause something to decrease. And that quantity is the Lyapunov function. And that has a very important consequence, right? Which is that the dynamics can't stop until you get to the minimum of the Lyapunov function. Now, for physicists, there's a much more natural way to talk about this, which is to say that this quantity, you'll notice I called it E and not L. It is a Lyapunov function, but it's also useful to think of it as an energy. And so what this means is that every possible state of the neural network can be assigned an energy. And this energy is fictitious, right? It's not a physical energy. It's a concept. And there's this landscape of energy as a function of all the different states of the neurons. And the dynamics of the network rolls downhill on this landscape. And so it ends whenever you're at a local minimum. And if there were noise in the system, you could imagine that instead of ending at the local minimum, then it could sort of float up and down a little bit in the same way when you have when a system is in thermal equilibrium. So why is this important? Well, suppose I ask you, sorry, let me pause for a second and look at the questions. I'm hoping that the slides are now updating. Someone asks about feed-forward networks is just more convenient for modeling. So I think the correct statement is that the initial idea is always that you simplify in the hopes of understanding something. And then you find out that the simpler thing that you created is actually useful in some way. Okay, so that's the order. So I don't think anybody would say that it's better to be feed-forward than to have feedback loops. I mean, better for what? On the other hand, it's much more understandable what's actually going to happen in a system that is feed-forward. The connection to the mathematicals and the physical energy function, I don't understand. So you should be careful, right? In many systems, when we say there's an energy function, we're actually referring to conservation of energy, which is exactly the opposite of what happens with the Lyapunov function. So Lyapunov function is characteristic of a system with typically dissipative dynamics. So the energy is always, the energy in this case is always decreasing. Okay, so yes, there's some discussion. let me say a few words about how the system can define itself in just a moment. So what Hopfield understood was that in this simple case of symmetric interactions, you have this energy function. So the dynamics of the network is that it's just rolling downhill. And then the question is, okay, how do I understand what the system computes? What can it do? And so the symmetry assumption in the matrix is a statement about the synaptic connections. So it's not that it's not that action potentials are running in the wrong direction. It's that if neuron I makes a synapse onto neuron J should make a synapse onto neuron I. Which happens sometimes, but it's just in the real brain, it's not exactly equal. So with this downhill dynamics, what Hopfield pointed out was that you could solve lots of interesting problems. For example, you could build a memory. So suppose that this energy function, instead of having one local minimum, has many local minima. Well, then in that case, depending on where I start, there'll be regions in the space of neural states that all get attracted back to the same local minima. And the idea was that this is like remembering something, right? I give you a cue, and there's many different cues I could give you, but then you always remember your friend's name, because all of these things make you think of your friend. So there's some state of the network that represents your friend's name, represents your friend generally, what they look like, the conversations you had, all sorts of stuff, including the name. And then if I get the network started somehow in the neighborhood of your friend, then the dynamics will restore the state that corresponds to your sort of prototypical image of your friend. Okay? And in particular, if this is going to work, you need to be able to program in the memories. You need to be able to store them. And so, for example, if you assume that actually, it would be easier to have this discussion not in terms of zeros and ones, but in terms of plus and minus ones. Same thing. If you imagine that the matrix Wij has this very simple form of being built as the kind of outer product of a vector, well, two vectors, but they're the same, so it comes out symmetric, then you can show that the minimum of E is when sigma vector is equal to Xi. And it's better than that because if you assume that there are a few of these, there are K of them, as long as K is much less than the total number of neurons, then what you can show is that the minimum energy is whenever your state of the network is one of these vectors. And so what that means is that you can actually write down how to program a network so that it always comes back to so I can take a network of a million neurons, or I can take 1,000 neurons and make sure that it always comes back to each of 25 things that I've stored. Furthermore, if I want to add something, you'll notice that the thing you need to add is something where the synapse that connects neuron I to neuron J, the thing you need to add is the state of neuron I and the state of neuron J. So that means that if I tell you that right now I want you to remember the state of the network, every synapse only needs to know what's happening on either side of it. It doesn't need to know what's happening on the other side of the network. And so that means you can have a rule or a learning rule for updating your synapses, which is completely local. Furthermore, the form of that rule is that you should look for correlations between the presynaptic cell I and the postsynaptic cell J. So one of the reasons why Hopfield's work had such impact was that not long before this, it had been discovered that if you take two neurons and you measure the strength of the synapse between them, then that strength of that synapse changes over time, and the way in which it changes is related to the correlation between the activity of the pre and postsynaptic neuron. And that's called Hebb's rule. Now, Hebb's rule was written down by Hebb in the 1940s, but it wasn't until the late 1970s that people actually were able to do experiments on single neurons to show that there's pairs of neurons to show that this actually happened. So there's more. You can think about, for example, programming networks of neurons to solve other problems. So basically, lots of the problems that you think the brain needs to solve, for example, when you look out at an image and you decide which pixels in the image go with which objects, you can think of that as being a kind of optimization problem. There's some term that says that pixels that are next to each other should be in the same object. There's another term that says that if there's a big gradient in the light intensity, then it's okay for the neurons, for the pixels to be in different objects, and so on. The boundary between two objects should be relatively straight. So if you end up drawing a zigzag boundary, then you should be penalized for doing that. So the problem, for example, of interpreting an image can be given a formulation as minimizing a function. But then in that case, I should be able to find some way of encoding that into this network of neurons, and I can compute whatever I want. In fact, you can do this with classical optimization problems in computer science. So the traveling salesman problem or something like that. You can think about formulating in this language. And so it was at this moment also that people started to think about the idea that you could take classical problems in computer science, give them a kind of physics formulation in which what you're doing is finding the minimum of some energy function. And then you could do statistical mechanics and ask, what is the nature, presumably if I work at high temperatures, I don't do a very good job of minimizing the energy. And then as I cool the system down, I drop into the local minima of the energy, which are the solutions to my computational problem. And so that suggests two things. One is that you could actually solve problems in that way by doing Monte Carlo and lowering the temperature, and that's called simulated annealing. And it also suggests that some of the analytic tools of statistical mechanics would allow you to compute solutions to these hard optimization problems of computer science, and maybe even suggest new algorithms for solving them and help you understand why it is that some problems are hard and some problems are easy. So Hopfield's work came at this nexus of developments in statistical mechanics, computer science, and neuroscience, and really had a huge impact because of that. So these are examples of the kinds of simple models that people have written down, and I hope give you some flavor for what they can do. And I think that at this point, we should pause for questions. Let me see. Right. So I think we're at the point where we're about to have, right. So we talked about the simple models. We then shifted to doing things on the board. And let's take a few minutes for questions, and then we will take a 10-minute break. So perhaps you need something to be true about the eigenvalues, about the list and unities, and figuring out. Remember that so I think not, although we could have a sorry, let me see if let me go back. If the connections are not symmetric, can you still make an energy function? So in general, no. In particular, if I have non-symmetric connections, then it's possible that the dynamics end up in an oscillation or in some sort of orbit, right? And they never actually stop. So even in the absence of noise. So part of the simplification here is that you only get dynamics that leads you to an endpoint, to a fixed point. If there are many local minima, could I get different names for the same object so initial conditions become way too important? So again, what's crucial here is not just that you have lots of local minima, but that they be programmable. You have to have a way for the network to learn where it wants to put the local minima. And so that's this business of using the outer products of vectors, which relates to Hebb's rule. In the roughest of senses, could train of thought be considered analogous to hopping from one of those minimum to another? Yes. And in fact, there are models of that flavor that came after Hopfield. For Hopfield's Lyapunov result, do you need something to be true about the eigenvalues of W? I don't think so. I think that if you have a symmetric matrix, then that takes care of it. But we could have a longer conversation. How big is the time constant for the network to reach its minimum? Nothing that I have told you so far prevents the possibility that to get to the minimum would take infinitely long if the network were big, or rather take a time proportional to the size of the network. In the simple patterns of connections that Hopfield considered, that tends not to happen. But if you made arbitrary connections, that would happen. It would take infinitely long for the system for a large system to relax. So again, part of the point about being able to sort of program the network is to be sure that nothing bad happens with the dynamics. And you could still study asymmetric cases, absolutely. This was meant, so the people who are asking about avoiding spurious spin glass states and things like that, they know much more than I was assuming you know for the purpose of this introduction. So this is all very interesting, but I wanted to just give you a flavor for where the subject started now really almost 40 years ago. If there's noise, is it helpful to literally think of the system as exploring a landscape? In fact, there are simple versions of introducing the noise in which instead of the dynamics coming to a stop, what you end up is with a stationary probability distribution, and that distribution will be essentially the Boltzmann distribution with the energy function playing the role of the energy, which is why statistical mechanics. So, you know, you can think about statistical mechanics in terms of the statistics of local minima of the energy function, but you can also, of course, think about the probability distribution over all the states, and you will get something Boltzmann-like. So it is noon. Let me suggest that we pause until 12:10. And we will reconvene, and I'll start in on the rest of the lecture for the day. Okay, so I'm gonna go get myself a cup of coffee. I recommend you do something similar. And I will be back in 7 minutes. Started again. Let me take one question that's left over from the first part, which is about graded potential neurons as opposed to neurons that have discrete on-off states. So Hopfield wrote this famous paper in 1982, which kind of touched off this revolution. There's a follow-up paper 2 years later, which actually has the title that graded potential neurons have the same behavior as discrete neurons. So, so the answer is emphatically yes. And one of the pioneering papers is addressed precisely to that question. Obviously, you have to sharpen the question a bit in order to give a meaningful answer. But yes, great question, and has the positive answer. So let me now talk a bit about how you record from neurons. So what we said was that neurons generate action potentials. And I talked about the action potential as the voltage difference between the inside and the outside of the cell. So in order to measure that, you actually have to put something inside the cell or something in the membrane. And that's hard to do. But fortunately, all of the neurons are bathed in a fluid, mostly water, but in particular, it's salty water. And so that means that currents that are flowing in and out of the cell will produce voltage changes in the fluid some distance away. And so you can pick those up without even actually touching the cell. And if you want a demonstration of this, you can put your fist in a bucket of water with two large metal plates and take leads from the metal plates and send them to an amplifier and a loudspeaker. And as you clench the muscles in your fist, you will hear the crackling of the action potentials arriving at your muscles. Okay, so obviously that doesn't allow you to measure what the individual neurons are doing, but you realize that you don't have to be inside the neuron. In fact, you don't even necessarily need to be all that close in order to pick up the signal. So the way this was used for many years is to put one electrode into the brain and bring it as close as possible to one neuron. And then you can be reasonably sure that you're picking up the signals that come from that neuron. And furthermore, you can recognize them because all of the action potentials will look alike. Not exactly true. There's some variation, but we understand that. So let me stick with the simple case. They all look alike. The problem is that that allows you to study one neuron at a time. But what we've been talking about is behaviors in the whole network that result from the interactions among large numbers of neurons. So the Hopfield model that I just wrote for you doesn't do anything interesting if there are only two neurons, right? It's interesting when the number of neurons is large and you can store lots of memories and so on. So how do you record from many neurons at once? I think that one of the places where this got the farthest first was in the retina, which is maybe not the most interesting example, but it has the wonderful feature the retina is approximately flat. So if you dissect the retina out of an animal, you can build an array of electrodes and put the retina down on the array and record from many neurons simultaneously. And what you see here are two generations of this sort of experiment. I particularly like this figure. What's happened is that you can see the green things are, the round parts are the cell bodies of neurons in the retina. The long tracks are places where the axons of many neurons are bundling together. And in fact, you can see the bundling is getting more dense as you go down in the figure. And that's because eventually all the bundles come together and leave your eye and go to the brain. What you can also see is the black spots, which are the electrodes on a piece of glass with wires leading away to the electronics to do the recording. And you can see that this particular experiment was set up to play with having electrodes with different spacing between each other and different sizes of the electrodes. So in the center, you have small electrodes that are very close together, and in the distance, you have electrodes that are larger and farther apart. And in particular, as you get near the center, you can see that if you count the black dots and you count the green dots, they're almost the same number. So you can fit almost as many electrodes as you have neurons. And you think that maybe you'll get lucky and you'll be able to record from essentially all of the neurons. So after some number of years, this experiment, this device becomes this one, where you have this sort of pincushion-like thing, or it looks like a pincushion, where there's now 250 electrodes, which are all at the same density as the dense ones in the middle here. And what you see here is a record from 2, 4, 6, 2, 4, 5, from 30 of these electrodes. And there are actually two colors on top of each other. So its voltage is a function of time from 30 of these electrodes with the retina sitting on top of it. And in one color is the actual voltage, I think in blue, and in the sort of yellowish or gold color is a reconstruction in which you assume that what you're seeing is there's some number of neurons, and every time one neuron generates an action potential, it produces a characteristic blip that appears on each individual electrode. And each electrode picks up signals from many neurons, and each neuron shows up on many electrodes. So there's a kind of complicated applied math problem you have to do in order to disentangle all this. But the fact that you can do it is illustrated by the fact that the yellow and blue curves are on top of each other. And so this is some of the history of that work. Now, I'm not actually going to talk about the retina, but it was very important for my colleagues and I because conveniently, Michael Berry, who was the one who in whose lab these electrode arrays were built, was in the next building. And we had this great collaboration where we started working, trying to use ideas from statistical mechanics to understand what these populations of neurons were doing. When we started, we were talking to them about maybe 10 neurons at once. Over the course of a decade, we got to the point where we were talking about 160 neurons and really being able to write down models for what is the probability that those 160 neurons will be in any particular state. And those got to be accurate enough that you could really take them seriously as statistical mechanics problems and ask questions like, where in the phase diagram of possible networks do you find the real networks? So this was very important background for us. And so I wanted to put it here also as an acknowledgement to the impact that our experimental collaborators had on us. What I want to talk about today is a very different kind of experiment. So instead of putting electrodes into the brain, you can do something else, which is get the neurons to report to you about what they're doing. So you genetically engineer an animal so that the neurons that you're interested in express a protein, and that protein is fluorescent, but its fluorescence is sensitive somehow to the electrical activity of the neuron. So the thing you really want to do is get that protein into the membrane and have its fluorescence depend on the voltage across the membrane. So you're directly reading out the electrical activity. There are proteins that will do that, but they're not quite right yet, okay? The signals aren't so clean. The next best thing is to get a protein that lives inside the cell, and its fluorescence changes depending on the calcium concentration inside the cell. Because every time there's an action potential, calcium comes into the cell. And calcium is a sort of common messenger for various biochemical things. So it's easy to get proteins to respond to calcium. So you have to synthesize a protein that puts together pieces of known calcium sensing proteins and known fluorescent proteins. And you end up with something that will sort of report to you about the internal calcium concentration, which follows the electrical activity. So first you have to do the genetic engineering. Then you have to be able to look into the animal to actually see what's going on. So you've turned the problem of recording from neurons into a problem of looking under the microscope. Here you're looking into a mouse, and you're looking into the hippocampus in a region called CA1, where the neurons come in roughly one layer. So that's a relatively easy case, right? You don't have to worry about the three-dimensionality. You can have the things in one plane of focus, and you see the neurons flickering on and off as the animal is doing its thing. There are many places, including the hippocampus, where neurons don't do anything interesting unless the animal is actually behaving. So if the animal's just sitting there, and worse yet, if it's asleep, nothing happens. So the animal has to be able to move around and do stuff. So you could think about building a sort of microscope that the animal carries around with it. Remember, these cells are 10 microns across, and in order to see the boundaries, of course, you need much higher resolution. So you really want to do very high resolution microscopy. And for those of you who are interested in these kinds of experiments, you should know that in this particular case, the way it's done is with scanning 2-photon microscopy. So you have a very intense pulse laser, which forms a very tiny spot. And that excites the molecules, the proteins that are in that particular spot. And then you collect all the light that comes out. And that tells you how much fluorescence is coming from that spot, which is related to the calcium concentration. And then you scan your spot around. So this image is not, you can't look under the microscope and see this. You actually have to synthesize it from the scanned. Incredibly sophisticated, okay? There's a lot of physics that goes into being able to build a microscope like this. But you'd also like to do this in an animal that's actually doing stuff, which means that it's running around. So how do you do that? Well, one idea is to get the animal to carry the microscope around with it. There are versions of that idea. The other idea is that after all, you don't care about, you're not trying to look at the whole animal, you're only trying to look at its brain. So what you do is you take the animal and you hold it down by the head so you can see in. Well, you have to open this up and you can see in. But then the animal is free to run, and he's running on a styrofoam ball which is suspended or which is supported by a column of air. So that means that he can run as he would in the wild. He can't move his head, but he can run with his feet. And you then monitor the motion of the ball, right? Since he's standing still, the ball moves underneath him. And you monitor that using a different kind of mouse. And by seeing how the ball rotates, you can compute what path the animal would have taken if you were running in real space. You can then use that to drive virtual reality for the mouse. So this is a kind of spectacular experiment that happens in my colleague David Tank's lab, where you're sort of pushing on many things at once, right? You have to get the genetic engineering right, you have to get the physics of the microscope right, you have to get the sort of computer engineering of the virtual reality right. And then you can actually watch what a large population of neurons are doing while the animal's running around and doing stuff. And what it's doing is controlled by you because you get to decide what sort of virtual reality setup you have. And there's a lot to be said about how to choose that, which I'm not going to talk about, because I'd really like to talk about how to think about the neurons. And a really important feature of these kinds of experiments is that the signals are incredibly clean. So what's been done here is to take that picture that I just showed you, right? Right? If you look at this, what you now want to do is to draw a little box around one of these neurons, average over that space, and plot the fluorescence intensity as a function of time. And then you get things that look like this. And so the signal to noise ratio here is really extraordinary, right? The fluorescence basically has a flat baseline with a little tiny bit of noise. And then every once in a while, the neuron is active, and you get a fluorescence signal that pops up for a second or two. You'll notice that this is slow, and that's because we're monitoring the calcium concentration, not the actual electrical activity. So you give up something by looking at calcium, but what you get in return is you can see all these neurons at once. You can think about the signal either as the continuous signal, which I'll call X later, or you can decide to discretize it yourself and say that the neuron is either on or off. And that's a mostly simple criteria. And you could just say if you're above a threshold, you're on. It's a little more complicated than that because you know something about how these fluorescent molecules work. And if you see, you notice that you know, when the cell turns off, the fluorescence decays relatively rapidly. So the fact that you saw it go down suggests that the cell turned off, but then the fact that it went down more slowly suggests that it came back on. And so you can do a little bit more sophisticated signal processing to do the discretization, but you can also think about the continuous signal. So the thing I want to emphasize is the extremely high quality of the data, which is really a tribute to my experimental collaborators. Quite extraordinary. Not all of these. You will see in the literature lots of experiments which say we recorded from thousands of neurons using these optical methods. Not all of the data looks like this. Okay. So it's really a very beautiful experiment. So that's the idea of how you record from neurons. You can either do it by directly monitoring the electrical activity, or you can do this indirect thing of monitoring calcium concentration using fluorescence. And so if that's reasonably clear, are the time axes really in seconds? In this case, yes. So that tells you that underneath these peaks, there's probably more than one action potential, right? You're probably seeing a burst of action potentials. And you'll notice that unlike action potentials, these come in different sizes. And that's probably because sometimes there's a burst of 2 action potentials and sometimes there's a burst of 4 action potentials and so on, right? If you want to, you can ask about how to deconvolve these signals to discover the underlying discrete events. The other thing you can do is wait for the molecules to get better so that they respond more quickly. And I think that that will actually happen relatively soon. So this is a subject under constant development. So what I wanted to talk about next is how do you think about these data? So we're looking at a population of more than 1,000 neurons. And remember, one of the ideas for how to approach these complex systems was that things would simplify in the same way that they simplify in problems that we understand in physics, that as you pass from a microscopic description to a macroscopic description, things would somehow get simpler. And so let's pause then for a moment and remember why it is that simple models actually work in physics. So in the interaction between physics and biology, physicists, of course, are much enamored of simplified models. And biologists are very suspicious. And they're right to be suspicious. And I think that some of the problem arises because as physicists, we haven't always explained very clearly where our prejudice for simple models comes from. And so there are many situations in which we completely understand how it is that simple models work, and we understand what work means in this context. So the example that I mentioned when I introduced the lectures was that if you think about the molecular dynamics of a fluid, and you think about fluid mechanics, there are as many different molecular dynamics scenarios as there are, or molecular dynamics models, as there are molecules that you can make fluids out of. It's endlessly varied. On the other hand, there's a very large class of fluids that are all described by the same equations, the Navier-Stokes equations. And the Navier-Stokes equations don't have very many parameters in them. They certainly don't have as many parameters as you would need to describe the force fields of molecules interacting with each other. So somehow, in going from what molecules are doing to what the macroscopic fluid is doing, things got simpler. And how does that work? Well, the dynamical case of molecular dynamics and fluid mechanics is a little bit complicated. So let me describe this in the case of equilibrium statistical mechanics. So if I think about equilibrium statistical mechanics, then what I'm doing is I'm writing down the probability distribution for a whole bunch of variables, call it xi, and the form of the probability distribution is that there's an energy function, right? And the probability distribution is given by the Boltzmann distribution. So what does the energy function look like? Well, typically what happens is that there are local interactions. So for example, it could be that your variables live on a lattice and the x's live on the lattice points. And if you look inside the energy function, you find that these guys interact with their neighbors, and maybe they interact a little bit with their slightly more distant neighbors, but that's about it. And so what that means is, for example, if you think about a simple magnet, you could say that you have magnets on the atomic scale, which are spins, sigma I, which are +1 or -1, that is to say they can point up or down. And then you write down an energy function which says, well, if the two spins which are next to each other point in the same direction, then that lowers the energy. And that's true for all the neighbors. And that model has a name. It's called the Ising model. And again, it says that basically if you see this, that has energy minus J. And if you see that, it has energy plus J. Okay? So this is the favored configuration. And what you could say is that because locally the spins want to point in the same direction, eventually, if all goes well, all of the spins will more or less point in the same direction. And that will give you a macroscopic magnetization, right? And that's how a magnet works. And in fact, we know that if you use the Ising model, you can predict the properties of real magnets. And actually, you can do something else, which is you can say that spin up is that I have a molecule in a small box, and spin down is the box is empty. And these are called lattice gases. And you can actually predict aspects of the gas-liquid transition using this model. And in fact, if you take this analogy seriously, it says that if you measure the right thing about a magnet and the right thing about a gas-liquid phase transition, they should look the same. And that's an experimental question, and the answer is yes, they do look the same. Whether this is the right model or not, they look the same. In fact, it's better than that, right? If you look near the critical point, that is to say, you go to the temperature where if you heat the magnet up a little bit more, it stops being a magnet. Or you go to the temperature where the density difference between the gas and the liquid vanish, then the behavior in the neighborhood of that critical point is quantitatively predicted by this Ising model. And in fact, the only parameter in this model is J, but that sets the scale of energy, which tells you what the actual value of the critical temperature will be. So in fact, there is no parameter, right? Nobody tells you what units to measure temperature in, so you could call the critical temperature 1. And all of the behavior in the neighborhood of the critical point comes out of this model correctly, quantitatively, to 3 decimal places, despite the fact that there is no real magnet that looks like this if you look on the atomic scale. So it's really important to understand that the reason that the Ising model describes real magnets is not because real magnetic materials are this simple. So why does it work? The answer is that it works because if you think about a model of a magnet and you say, well, look, I don't really care what's happening on the small scale. I'm interested in what happens on larger scales. So what I could do, for instance, is to say that instead of trying to describe a magnet where I look at every single atom and ask what its spin is doing, what I could do is to look at blocks. So here, for example, you'll notice that I have 1, 2, 3, 4, 5, 6, 7 spins up. So what I'm going to do is I'm going to replace that block by 1 spin that's up. And then I'm going to do the same thing over here. 1, 2, 3, 4, 5, 6. That also has a majority up. 1, 2, 3, 4, 5, 6, 7. I didn't do a very good job of this. That one also has a majority that's up. 1, 2, 3, 4. Maybe I can cheat a little bit. And this now, thankfully, has majority down. And what I can ask is, if I wrote down the energy as a function of all the spins, and these are the microscopic spins, and they're on some scale, which I could call 1, right? Let's call that distance 1. These objects I can still write the energy as a function of these new spins, right? I guess to be helpful, maybe I should write this. The energy is a function of the sigma I, which are red objects. And here, they're functions of the green object. And when I do this, right, I can always do this because the energy, right, every state on this side is some combination of states over here, right? In order for this spin to be up, there has to be a majority of up over here. And so you can check and you can sum up over all the different ways of arranging these spins to give you this spin up. And then when you compute the probability in the Boltzmann distribution, you just sum over all those states to give you this new state. Right? And the question is, what's the form of this function? So let's say you started over here with the simple Ising model where everybody just talks to their neighbors. But now when you go to here, right, these spins will interact with each other. But even if the spins that you started with only interact with their nearest neighbors, probably these will interact with their next nearest neighbors. And in fact, there'll be more complicated interactions where all four spins interact with each other in some complicated way, right? So what you can think about is that what you're doing here is you're starting with a description at the shortest length scale, and then you're averaging over the details at the short distance and kind of zooming out. And in the process of zooming out, you can rewrite your description of the system. And in this simple case, the variables are the same. You started with spins that are up or down, and you have spins that are up or down. And you can do it again and go to a longer length scale and a longer length scale and a longer length scale. And as you do that, you can always put the model back in the same form where spins talk to their neighbors, they talk to their next neighbors, they talk to each other in pairs, they talk to each other in quadruplets and so on. And you can take all of the terms that you generate. And what's important to realize is that even if you start with a simple model at the shortest scale, here, over here, at the first step, it'll actually get more complicated. So that means you could have put more stuff into here. You could put in more terms. And so you generate a whole set of terms over here. And what's surprising is that when you do this, if you do it again and again and again, most of the terms that you might imagine putting into the model eventually get smaller as you go out to longer and longer length scales. And what that means is that if you want to describe physics on long length scales, you can forget about most of the terms. Okay? And so that's the spirit of why it is that something like fluid mechanics can be simpler than molecular dynamics, and why it is that the Ising model, which is incredibly simple, can describe a real magnet, especially near the critical point. And why near the critical point? Well, near the critical point, the fluctuations in the internal configuration are correlated over very long distances. And so all the physics is happening on long length scales. And so that means that that's really what you care about. And so you want to coarse grain in this sense from the microscopic all the way to the macroscopic. And so it is a spectacular result of this process, which is called the renormalization group, that the models tend to get simpler rather than more complicated. So let me make one observation, which is that if I write the energy is, you know, there's a sigma I, sigma j, maybe there's a sigma I, sigma j, sigma k, sigma L, maybe there's lots more terms, and there's a j for the nearest neighbor, there's a j1 for the nearest neighbors, there's a j2 for next nearest neighbors, and so on. So every one of these terms, as I go from scale L to scale, let's say 2L, every one of these j's goes to some function of what the jn's were, and in fact, they all mix together in some complicated way. But the point is that if you look in the space of all the so this is the space of all the parameters of the energy function. It's the space of these j's. Then what you find is that there are these interesting trajectories where if you follow, you find that, for instance, along certain directions, you go one way, but if you're on the other side, you go the other way. You might also turn off into other directions. And what you realize is this means that as you go from the microscopic scale to the macroscopic scale, it doesn't really matter where you start, it matters where you end up. So for instance, if you start over here somewhere, then you're going to end up off in this upper right-hand corner. And at that point, the precise value of the parameters along this axis don't matter anymore. What matters is the value along this axis. So some parameter has basically been squeezed down, and there's another parameter which is important. And really, this is in some very high dimensional space. And what's important is that most of the parameters are shrinking to zero, and a few of them have a chance of becoming large. What you also notice is that something special could happen, which is that you could end up at a place where the parameters don't change at all. And let me remind you that there's a familiar, for those of you who don't know about this, there's a familiar example of this, which is that if you just take random variables that are independent of each other and you start averaging them together, then you know that eventually the probability distribution becomes Gaussian. That's the central limit theorem, as long as the moments are finite. So that tells you that if you give a probability distribution of a whole bunch of variables and you start averaging them together, the distribution can get simpler. But the case that's familiar is the one where you just become Gaussian. What the renormalization group shows you is that you can become simpler, but not trivial in that Gaussian sense. Okay, this is not a substitute for a whole course about the renormalization group, but I hope it's an introduction to these ideas. And I hope it dispels once and for all the idea that simple models work in describing real materials because materials are simple. That's not true. They work because you're asking questions about macroscopic behavior, and the renormalization group drives you towards simpler behaviors on the macroscopic scale. So the question is, can we use these ideas to think about neurons? And so we developed these, my colleagues and I developed these ideas in a series of papers. And as I say, not only will the slides be on the website, but I think we'll also be able to give you copies of the papers. So how are we going to do this? Well, what we did in the case of the magnet was we averaged a spin with its neighbors. But with neurons, what's a neighbor? It's complicated, right? Because neurons can connect to each other in very complicated ways. So your spatial neighbor is not a very good guide. So instead, what you could do is just ask, your neighbor is the neuron that's most correlated with you. So what we do is we look through all of the neurons. We find pairs of neurons. We find the most correlated pair of neurons, and we average those together. And then the next most correlated pair, and we average those together. And we do that until we're finished and we have half as many neurons as we had before. And then we do it again, and we do it again, and we do it again. And so this is like what we were doing with the blocks of spins in the Ising model. Okay, how do you tell what's happening? I mean, you can do this if you want to, but what do you do? So it turns out that the question of how you analyze the data was addressed when people started using these ideas to look at Monte Carlo simulations. So what you're interested in is the whole probability distribution, but that's very hard to get at because it's a high dimensional object. So what Binder proposed a long time ago was that you should look just at the local variables. So if you're averaging together spins, what you get is a kind of local magnetization. So you should look at the distribution of the local magnetization. So in this case, right, we have this continuous variable, which is the fluorescence signal we get from every neuron. And that value, you remember, it can be really zero because the signal to noise ratio is so high that you can tell when the neuron's completely silent. And there's some probability that the activity is zero. And as you average the neurons together, you preserve some probability that the activity of that sort of super neuron is zero. So that's here. And then for the non-zero things, there's some probability distribution. So you can track these. And what's astonishing is that if you look at this, let's actually look at this one first. So we're looking at the probability distribution of the non-zero values. And what you see is that you start with something funny looking. And then as you keep rescaling and grouping together more and more neurons, the probability distribution just stops changing. It takes on some very characteristic form, which is almost an exponential distribution, not a Gaussian distribution. Furthermore, if you look at the probability that this group of K neurons that you've blocked together is silent, if the neurons were independent of each other, you'd expect that the probability falls exponentially because you just keep multiplying the probabilities that each neuron is silent. And instead, what you get is this kind of stretched exponential with an exponent, which has, well, first of all, you can see that the behavior is very accurate across more than 2 decades. And the exponent is determined to 1% precision. But what's astonishing is if you do the experiment in 3 completely different mice, you get the same behavior also with 1% precision. So this tells you that there's something going on in this network that reflects this scaling and coarse graining behavior that's incredibly reproducible across animals. Let me focus on something that is, since time is getting short, let me just focus over here. In the physics problem where you put blocks of spins to bigger and bigger blocks of spins together, your intuition is that it takes, one spin can flip very easily, but for a whole block of spins to turn over is harder, right? Because it's a bigger block. So what you expect is that the dynamics get slower as you make your blocks bigger and bigger. But the dynamics might stay, there's a possibility if you're following one of these simple trajectories in the normalization group that the form of the dynamics stays the same. And that's what you see here if you look at the correlations in time of blocks of different numbers of neurons. They do indeed get slower, but if you rescale by the correlation time, then they look exactly the same. And furthermore, that correlation time goes as a power of the cluster size. So why are there these power laws? It's again, just like in the example of the central limit theorem. Remember that as you approach the central limit theorem behavior, the mean stays constant and the variance goes down as 1 over the square root of the number of things that you're averaging together. So standard deviation goes as 1 over the square root. So that square root behavior is, right, there isn't some magic scale when you average things together, the variance drops. It keeps changing and changes in this power law way. So part of the point about the renormalization group approach to these fixed points is that you get, again, power law scaling with how large a block you put together. And that power law is not trivial in the way it would be if you were approaching a Gaussian. I don't have time to talk about this. So I'll note that I've talked to you about this as if it were a physics problem. I'm just coarse graining. I'm doing the same thing I would in the Ising model, trying to see if something simple happens. But then you should worry because, well, this really is a network of neurons, and that we know some things about this particular network of neurons, and maybe that has something to do with what we're seeing. So you can sort of chase down all of these questions. And trust me, none of these things are the essence of what's going on. It really seems as if when you coarse grain the neurons in this region of the brain, you actually see this orderly behavior where the statistics of what individual neurons are doing is complicated, but as you block them together, it becomes simpler and simpler, and in fact seems to approach some probability distribution which is fixed and isn't the trivial one of being Gaussian, right? And along the trajectory to that, you see these power law behaviors, which are exactly the kinds of things you see at a critical point in the physical case. And furthermore, in equilibrium statistical mechanics, and furthermore, the exponents that characterize the motion along those trajectories are extraordinarily precise and reproducible across experiments. So this is all phenomenology, and it's all in the spirit of trying to convince you that it is possible for simple things about this complicated system to emerge as you go from the microscopic to the macroscopic. And in fact, it emerges in this case in exactly the same way that we do it in the physics problems that we understand. So this is offered as an example of this approach of trying to use emergence as a way of cutting through the complexity of the biological system. So we have about 10 minutes. And what I'd like to do is take some questions about what I've said, but I would also like to leave some time for somewhat more general discussion if people have questions about how you find your way in this field and so on. So let me start by answering questions that are in the chat, and I'm going to be quick about it. If you start with without diagonal interactions, why do you get diagonal interactions once you block? I invite you to do the calculation. You'll see that that happens. It's because there's nothing special about your nearest neighbor anymore. The neighbors that you pick last kind of far apart. So I was showing you a sketch of neurons with varying degrees of correlation. And so you should be careful, right? Slightly tangential, being a comment on the critical brain hypothesis. So there are the idea that biological systems might be tuned to a critical point has come up in many different contexts, including the one that's referred to by Damian Cho. There are dynamical notions of criticality, there are statistical notions of criticality, and what you're seeing here is criticality precisely in the sense of the normalization group, which is that you have an invariance under coarse graining, which I think is the strongest one. It is true that all the systems we understand, right, there is some underlying effective field theory that should describe them. And of course, we would love to construct that. I find it interesting, and this is not meant as a criticism of the person asking this question, but it's fascinating to me how quickly people will go from why on earth would you think that the brain is at a critical point to can you tell me the field theory that describes the underlying critical point? I think that maybe our desire to make that jump is so strong that we're often not as rigorous as we should be about the experimental evidence. And so I think I've focused here really on the data because I think that's what's changed in the last decade is the possibility of analyzing data in this way. It was the quality of data that makes it possible to answer these kinds of questions. And so I think it sharpened, that looking closely at the data sharpens our theoretical questions. And I agree that that is the interesting theoretical question. Can you tell me what is the underlying theory that generates this behavior? But of course, if you didn't go through this, and I should say, I've glossed over all the things you should check to be sure that you're not fooling yourself, which are many. And there's a short paper, but there's also a long paper, which by the time it's published will be even longer. It's currently on the archive. So there's a lot of things that you have to convince yourself. So do you predict that there are phase transitions? So what's interesting is that what we're seeing is the behavior of a system which is poised at or near its critical point. What's not so obvious is, if you will, what's on the other side of the critical point, right? What would you be transitioning into if you went further? There are people who have tried to address that question in a variety of experimental systems, but I think certainly I don't know the answer in this system. Are there examples where coarse graining of another parameter other than space or equivalently energy? So I described the coarse graining process as if you were doing it in real space. You average with your neighbors. Another way to do it is to Fourier transform and filter out the short wavelength components and then transform back. That's called momentum space or momentum shell normalization. And we've also done, we've argued that there is a way of doing that in the neural context as well. It's important that in our context, the procedures we're following are analogous to doing it in real space or doing it in momentum space. But importantly, in neither case is it actually space or momentum, right? The wavelength, which is the variable we use, because that's not the right quantity. You're right. You're not in a system that is translationally invariant, and you're not in a system that has local interactions. So how should you understand emergence? Let me hold that for a moment. Let me get the technical things first. Does the exponent measure anything about animal status or behavior? So what we know is that these exponents are incredibly reproducible if the animal is doing the same kind of behavior. What we don't know about yet is what happens if you're doing different behaviors. What are good resources for learning about the renormalization group? When we post the slides, I will add references for sort of textbook level things. Is there a sign of relevant perturbations that can detune? Sorry, wait a minute. Is there a link between the structure of evolution and the emergence of biological criticality? The next question is whether there are relevant perturbations. These all revolve around whether I can take all the mathematics that I understand surrounding criticality and the normalization group and connect it to something that actually matters for the biology, right? So you'll notice that in this part, the things which emerge are behaviors of the network as a whole, but I haven't told you why the animal would care about any of those things. If I think about the example of a flock of birds, then the thing that emerges is that all the birds are flying in the same direction, and the notion of criticality has to do with long-range correlations between the fluctuations and what the birds are doing and so on. And so there, it feels like I'm closer to being able to say something about why this matters for the organism. In the case of networks of neurons, there's lots of words that are written down about why criticality would be a good thing, but I actually don't find any of them very compelling. And so we've sort of resisted doing that. But I think that's an essential point, is we eventually have to figure out, you know, when I talked about the embryo, I talked about optimization and the idea of squeezing out as much information as you can in order to do something that the animal actually cares about. Whereas here, we're just being completely phenomenological and not sort of worrying about what's important for the animal. And somehow these views have to get linked. And in fact, that's sort of the next question, which is about what's the link between these renormalization group ideas and the transmission of information, or maybe even the transmission of semantic rather than syntactic information. The fantasy would be that, you know, right, in some way, semantics happens on a larger scale than syntax, right? So you'd like to have a renormalization group approach that goes from, you know, phonemes to syllables to words to phrases to ideas, right? And we don't know how to do that. Do you have thoughts about mechanism? Nothing useful to say in a short time. Are there methods to coarse graining things? Are there bad ways? So what I should say is that in the systems that we understand, right, in equilibrium statistical mechanics, there are many ways of implementing coarse graining. And there are some things, and the different ways of implementing coarse graining, if, for example, you're trying to use the results of Monte Carlo simulation to understand what's going on, then different ways of doing it are more or less efficient in the use of the data that you have available to you. But from an analytic point of view, they all arrive at the same answer. So for instance, if you think back to the simplest problem, which is averaging variables together to get Gaussian variables, right? You can average them together in different ways, and it doesn't matter. You always end up with the same Gaussian distribution. There simply isn't a canonically best way of doing coarse graining. And so what we've done, what I've told you about, is literally the first thing that we tried. And it worked in the sense that we uncovered orderly behavior. I don't believe that this is the best way to do it. And there are situations in which you're more or less guaranteed that this wouldn't work. And I guess the positive way of saying that is that there could be qualitatively different coarse graining approaches which would uncover different kinds of ordering or critical behavior. Do we think about graded potential networks in the same scheme? So someone also asked about what's on, what are the two phases? And I said, I don't know. I mean, we can work on it, but the short answer is I don't know. Can we think about graded potential networks using a similar scheme? So importantly, in some of this work, we treat the raw fluorescence signal as the variable characterizing what individual neurons are doing. So in that sense, you're taking seriously some continuous variation in the activity of the neurons. In other cases, we discretize. And the qualitative results are the same. I don't think there's any crisp relationship between emergence and local entropy. What about disentanglement of representations? Fantastic question. I don't know. What is the most relationship thinking insight to philosophy of science? If you're asking what aspects of what has been happening in our field do I think should be important for the philosophy of science, I could answer that question, although I don't know that it would just be my opinion. If you want to know what's been important for the philosophy of science, then you have to ask a philosopher of science. Sorry, I'm not trying to be flippant. It is important to remember that when you talk about the interactions between different fields, if you say that field A has an impact on field B, you kind of have to ask the people in field B what they think, right? So that's why I talk about the physics of biological systems and not what physics has taught us about biology. You have to ask a biologist what physics has taught about biology, because they're defining their questions in their own terms. I think people an important feature of this is that people who come from different intellectual traditions, they don't just have different tools for answering the same questions, they ask different questions. What about the strong top-down causation? I have to say that's something that I have not, I don't always understand what people mean by this. There is an obvious notion of top-down causation, which is that, look, evolutionary change happens at the molecular level, right? You get mutations. And then the impact of those mutations is felt. I mean, we say that you select for a mutation or against a mutation, but that's not true. What you select for is the behavior of the animal and whether it is correct in the context of its environment. And that's a very macroscopic thing. So in some way, what's characteristic of evolutionary dynamics is that you sort of go from the microscopic to the macroscopic, and then the hand of natural selection reaches down to the molecular level and crosses out certain things or amplifies them, right? And so in that sense, I don't know you're sort of guaranteed to have something that looks like top-down causation, right? Just by the nature of the dynamics. What about quantum mechanics and biology? I've been fascinated by this for all of my scientific life, and I think that the best current answer is that there are places where it seems unambiguously relevant, and whether it's broader than that is a hard question. Good. It was our plan to have some more general discussion about life in science. And I guess for this in particular, it might be fun to talk about directions. You know, how do you find your way if you come from a traditional physics background to working on problems that touch biological things? And I would be very happy to have that discussion, but I think we're sort of near the end here. And we still have the plan of scheduling one more, one extra lecture at the end, or one lecture period at the end, with as many of us as we can get in one place, so you guys can ask questions about well, whatever's on your mind, not so much the technical questions. I would be happy to spend 15 minutes doing the broader career things from my own point of view now, if people would like to stay. But I also feel like, people set aside blocks of time, and I worry a little bit about, people who need to do something else. So I would prefer that we schedule one more session and everybody is there, which I think gives a more equal opportunity. So let me then look forward to seeing you at that, and thank you for your attention. And as always, all of the lecture materials will find their way to the website actually quite soon, I suspect. So thank you all. Mm.